Another mechanism used by cells in order to transport substrate molecule is active transport. So active transport is against concentration gradient and it requires energy in the form of ATP or as electrochemical gradient, or sometimes light energy is used for transporting molecules. Now, this depends on a particular pump, and that is called the sodium potassium ATPase. The structure of sodium potassium ATPase has been elaborately studied. Now, there are different types of sodium potassium ATPase. There is a P type, F type, then ABC type. All these type of pumps has been observed in different cells. When we look at the concentration of these sodium ions, in the cytosolic side, the concentration of sodium ions is only about, let's say, 10 millimolar, and potassium ions is, this is in the cytosolic side, 10 millimolar, and potassium ions in the cytosolic side is somewhere around 100 millimolar. In the extracellular fluid, this concentration is in reverse. For example, sodium level is about 100 to 140 millimolar and potassium level is about 5 to 10 millimolar. So this concentration must be maintained in a cell. The sodium ions must be pumped out of the cell for the normal functioning of a cell. Now, how is it achieved? So this is achieved with the help of this sodium potassium ATPase. Sodium potassium ATPase, as we have seen, there are different types: F type, P type, V type, ABC type of sodium potassium ATPase. They all depend on ATP hydrolysis for activity. Roughly about 20 to 40% of the total metabolic energy is utilized by this sodium potassium ATPase. A neuron, it may go up to 70% of the metabolic energy is utilized by neurons for pumping or maintaining this ionic balance in a neuron. What is the main function of the sodium potassium ATPase? It will pump 3 sodium ions out of the cell and also 2 potassium ions. So, sodium potassium pump has been used for maintaining the concentration of many ions like sodium, potassium, magnesium, calcium. The second transport mechanism that we are going to look at is secondary active transport. So why it is called secondary active transport? It is because if the gradient generated from the primary active transport mechanism is the driving force for transportation of molecules. Secondary active transport is also known by co-transport. So in the secondary active transport, there are 2 kinds of secondary active transport has been observed. The first one is called symport mechanism, and the second one is called an antiport mechanism. What happens in a symport mechanism? So in a symport mechanism, if this is the substrate molecule which is outside, this molecule must be brought inside. So, in order to bring the substrate molecule in, ATP hydrolysis occurs. ATP is hydrolyzed to ADP and PI. That energy is utilized for bringing the molecule inside. This is a model for glucose or galactose transport across membrane. It has 2 binding sites, one for glucose and another one for sodium. It functions against concentration gradient. The carrier has low affinity for external glucose before sodium binds. After glucose binds, conformational change occurs and present glucose and sodium inside.
unloaded carrier undergoes conformational change and expose binding site to the external surface. The most interesting part is glucose entry gives a free energy of about 9.2 kilojoules per mole under cellular condition. This is because the external cellular condition for sodium is 143 millimolar and internal is 14 millimolar. This change, this difference creates a membrane potential of minus 50 millivolt. And this free energy generates a 66-fold concentration gradient for an uncharged glucose molecule. So the import is driven by a changing in the sodium concentration in the extracellular fluid.
When it comes to antiport mechanism, here is a substrate molecule which is outside, which must be brought inside, substrate in. Now, in order to bring this substrate molecule in, an electrochemical gradient is used, but at the same time, when this molecule is coming, another molecule will be transported outside. So, this is known as the antiport mechanism.
So antiport is a process where two different species of solutes or ions are pushed across a membrane but in opposite direction. One of these two species is allowed to move from a high concentration to a low concentration. We know that this will cause a kind of an entropic energy, and that energy is used to drive the movement of outer solute from a low concentration to a high concentration. Breakthrough about the antiport mechanism is by very famous biochemist Ephraim Racker in 1960. Ephraim Racker and his colleagues were looking at mitochondrial protein that could exchange inorganic phosphate for hydroxide ions. This protein is located in the inner mitochondrial membrane and transport phosphate ions for use in oxidative phosphorylation. So that is how these molecules or this antiport mechanism was originally discovered. Excellent example of an antiport mechanism is the sodium calcium antiporter, which was first reported in 1968 by Gerald Rotter et al. And this antiport system allow the entry of 3 sodium ions into cell in order to transport 1 calcium outside. And this has been observed in the heart muscles.
So in this video, we have looked at how the substrate molecule is playing a very crucial role in regulating metabolic pathways, and which are the ways by which the concentration of substrate molecule is regulated in a cell. So these are the aspects that we have seen in this particular video.